Good afternoon, and thank you for having me here today. My name is Youngeun, and I'm a PhD student at McGill University, and I'm working with Boris Bernhard, and I'm also working with Alan Evans. So today, I'll talking about the superficial white matter architecture on high-field MRI and histology. So the objectives of these presentations are like that. So first, to explore the methods used for superficial white matter mapping, and the second, to characterize the superficial white matter in high-field 7T MRI and histology. So before we proceed, let's first define the superficial white matter. The superficial white matter refers to the white matter which located directly underneath the gray matter, which containing U fibers that connect agents gyri. And these regions remain incompletely myelinated until later in life, suggesting its important role in brain development and maturation and aging and various disease studies. But this superficial white matter is not uniform. Its structure varies across the regions, bound by the gray matter and covering the gyri and neighboring circi. The superficial white matter consists of a mix of the fiber population with varying directions and destinations. So the dense packing of these superficial white matter fibers in the superficial white matter and the presence of multiple crossings and its proximity to the cortex, along with inter-regional and inter-individual variations that make in depicting this microanatomy highly challenging. And additionally, the complex 3D structure of superficial white matter bundles plays a key role in this area. So there are 97 pyramid-shaped crossings per lateral white matter surface, which are crucial in forming the neural network within the superficial white matter. So many studies have used these pyramid-shaped crossings to explore and illustrate the superficial white matter structure in the human brain. So in the superficial white matter study, understanding the myellar architecture is important, as local differences in the thickness and compactness of fiber layers, as well as the length and strength of radial bundles, allow us to identify distinct areas of myellar architecture organization. So integrating both myellar architecture and cytoarchitecture perspectives will enhance our understanding of this complex structure of the function of the superficial white matter. So in vivo, we can investigate this superficial white matter in living humans using MRI with various features. For example, iron and myelin are especially important contributors to MRI contrast in the superficial white matter. And additionally, the T1 and T2 weighted ratio can also provide insight into gradual changes in the microarchitecture properties of both gray and white matter. So to study this superficial white matter, several methodologies are currently available. For example, these include tractography-based method, and surface-based tracking method, and mask-based approaches, and Laplacian-based method. But these methods have some limitations. They are computationally expensive and challenging to implement it with multiple dependencies. And also, they are often not well documented and lack open source availability. So in our lab, my colleagues, Jordan De Cracker and Raul Crosses, and I developed a repository to solve a Laplacian equation from T1 weighted images and generate the surfaces at various superficial white matter depths based on this Laplacian field. And additionally, we also has integrated this script into this MiCaPy pipeline, an open processing framework for multimodal MRI dataset. And this pipeline built in Python and is designed to be open source and reproducible and with minimal dependencies. So you can run it independently or integrate it with the pipeline separately. So to characterize the superficial white matter in vivo, we are utilizing our 70 MRI data set, MICA PNI. Boris mentioned this before. And this data set includes 10 healthy subjects. And for this study, we are using 2 myelin-sensitive quantitative MRI. First is quantitative T1 relaxometry, T1 map, and second is magnetization transfer saturation, MTSAT. And for our ex vivo data set, we utilize 2 histological data sets. The first is BigBrain data set. Probably everyone's familiar with this data set. And I use 200 micrometers resolution of the data. And it comes from a 65-year-old man, and it's stained for cell bodies. And the second data set is a head data set with a 400 micrometer resolution. Although there are 2 subjects in this data set, but we are currently using only 1. And this data set includes multiple histological stains with 6 different staining methods. But for our study, we are using 2 stains method, Biotrovsky and pervalbumin, which target nerve fibers and interneurons respectively. So here is an overview of the data set used in our study. T1 map is sensitive to myelin, axon, and iron, all of which contribute to prolonged T1 relaxation times. And MTSAT is sensitive to myelin density. And the BigBrain dataset is stained for cell bodies using the Merker method. And the AHEAD dataset includes Biotrovsky and pervalbumin, which target nerve fibers and interneurons. So now I will briefly introduce our method. So to sample the superficial white matter surfaces from the quantitative MRI, we first solve the Laplace equation using the T1 weighted images. And from this, we extract 15 surfaces, in total of 15 surfaces ranging from 0 millimeter, the gray matter white matter interface, to 3 millimeter into the white matter. And then we map these surfaces onto each quantitative MRI, for example, T1 map and MTSAT. And here is an intensity profile from the MTSAT, for example. And the y-axis represents the white matter depth, and x-axis represents the surface vertices. And this profile explains the intensity transitions across this superficial white matter region. So we observed that the intensity profiles of T1 map and MTSAT vary along the depth of the superficial white matter. Both T1 map and MTSAT exhibit distinct intensity changes between 0 millimeter to 2 millimeter in depth, here and here, which may be related to the complexity of this area, this region. And below 2 millimeter, the intensity distributions in both MRI sequences become more stable. This stability might indicate a transition to a more homogeneous white matter myeloarchitecture and the emerge of deeper long-distance white matter fibers. And for the histology, we first get a tissue segmentation from histological feature and then solve the Laplacian field. Given the higher resolution of the histological dataset, we sampled 50 surfaces ranging from gray matter, white matter surfaces to 3 millimeter into the white matter. And these are the intensity profiles from the BigBrain and AHEAD dataset. Since BigBrain captures cell bodies, we observe distinct changes between 0 millimeter to 0.6 millimeter. But the nerve fibers and interneuron stains from the AHEAD dataset show more pronounced intensity changes between 0 millimeter to 2 millimeter in depth. And next, we examine intensity changes related to cytoarchitecture classes of gray matter, which describe the cellular composition and structural characteristic of cortical regions. The results varied by feature. For example, in vivo dataset, we observed a distinct shift in intensity profile patterns in the paralimbic here and insula area here, known for their diverse and heterogeneous cell types. And Biochovsky stainings also revealed notable differences in intensity and skewness across the classes. Importantly, all intensity profiles showed notable changes between 0 millimeter to 2 millimeter in depth. So for future study, we plan to investigate intensity changes from gray matter to superficial white matter by McGill architecture class. This connection between gray matter and superficial white matter may suggest that superficial white matter intensity changes are influenced by the intricate layering and cellular diversity of cortical regions, but also the 3D local architecture of the gyri. But currently, the variation in intensity profiles at different depths remains complex. So to better understand the superficial white matter architecture, we have implemented a method that combines superficial white matter covariance analysis with dimensional reduction. So first, these vertex-wise intensity profiles were cross-correlated, resulting in microstructural profile covariance matrices that represent vertex-specific similarity in each quantitative MRI and histology feature across the superficial white matter. And each average MPC matrix was converted to a normalized angle affinity matrix, and diffusion map embedding was applied. The eigenvalues of the embedding, also known as gradients, describe the variance across the superficial white matter MPC. So we implemented this method in the Mica pipe processing workflow software too, to study the complexity in superficial white matter covariance and can be applied to any quantitative MRI. The first gradient of T1 map clearly distinguishes between the main cerci and gyri, and when you plot the intensity changes according to each eigenvalues, distinct pattern emerge particularly after 1.2 millimeter. For empty set, the gradient showed similar intensity changes to T1 map, especially in central sulcus and insula regions, but it also includes the frontal blade. But the intensity changes by eigenvalue show marked differences between the superficial white matter and converge around 3 millimeter. And the histological gradients are more complex. The cell body stain shows significant intensity changes within the first 0.6 millimeter, with eigenvalues converging around 2 millimeter in depth. The nerve fiber stain reveals distinct intensity differences across the eigenvalues, with a big difference in skewness. While the interneuron stain shows that the eigenvalue stabilized at around 1.5 to 2 millimeter in depth. And lastly, we examined the relationship between the first superficial white matter gradient and the functional cortical degree centrality derived from the PNI dataset, mean resting state functional connectome. In this analysis, degree centrality indicates how directly connected a specific brain region is to various other regions, playing a crucial role in information integration and diverse brain functions. And all the first superficial white matter gradients showed significant correlation with this degree centrality, indicating a potential relationship between areas of critical functional importance and connectivity and superficial white matter architecture. So from this study, we can conclude that the deep white matter interface begins approximately 2 millimeter below the gray white matter boundary surface, and the structure organization of the superficial white matter is way more complex than what it thought, with 3D structure, multiple layer, local connection, and high density of crossing fibers. So superficial white matter similarity captured the complex structure organization in the superficial white matter area, with high variability in the transition area between superficial white matter and deep white matter in all modalities. So for future studies, superficial white matter informed tractography and connectome mapping should be considered. The endpoint should be the white matter surface with dynamic seeding between 0.2 millimeter to 2 millimeter. And additionally, high angle for each step is necessary for probabilistic cryptography algorithms, while deep white matter should be excluded. And thank you for all my colleagues and country collaborators, and feel free to get in touch. And sorry for taking too much time.